Good morning to one and all. Today we will discuss about the topic locomotion, excretory products and their elimination along with the locomotion. First, when you come to the excretion. Excretion means X means shifting out. Cre means not required by the body. Shifting out of the materials which are not required by our body out of the body is called as excretion. In the process of excretion, we can eliminate generally the nitrogenous waste. Based on the elimination of the nitrogenous waste from our body, whole animal kingdom is basically categorized into three parts. The first one is aminotelic, means ammonia excreting animals. You can see that the animals like aquatic amphibians, aquatic insects, and also the water living, most of the water living animals. Coming to the second type is uricotelic animals. Ureotelic animals, urea excreting animals, ureotelic animals. Their moderate water and amount of water is sent out of the body. And ureotelic animals, we can see that mammals, terrestrial amphibians and marine fishes, and which release the uric acid, one of the components which is generally least toxic, and as well as which also takes the water from the most of the food like the certainly like the lizards or the aerophilic animals. And here the excretory material may be as some solid pellets, also some cases. And generally when you come to the toxicity of the animal, based on the nitrogenous waste, generally NH, the ammonia containing animals waste is highly toxic, and here more water, they will eliminate also more water because they live in the aquatic conditions, and the uric acid is least toxic and the urea is medium toxic. Now coming to the this, in the case of the nitrogenous waste, kidneys are not required, but in the case of mammals, kidneys are present, is the structure and functional unit of excretion, and uric acid, it is a part of the food. Now coming on to the other most important thing, generally some amount of, always some amount of urea is retained in the kidneys to maintain the osmolarity. And when you come on to the some of the simple tubular excretory parts in the most of the invertebrates, and when you come to the other animals, complex tubular excretory parts are present in most of the cases, that is the vertebrates. And when you see the categorization of the organ present in the organisms, we can see that 5. So that is M, Malpighian tubules, P, protonephridia, P, and A, antrenal glands, C, coxal glands, K, metanephric kidneys or kidneys. So coming to this, you can remember that with the help of a name called as MPAC. So your Malpighian tubules, P, protonephridia, A, antennal glands, C, coxal glands, K, kidneys. So protonephridia is the primitive type of excretory organ, and it is starting from the platy and nemati element. These flame cells are the major excretory organs, where they look like flame, as they will be separating the excretory materials from the food by a tuft of cilia present in that, and it is present in the platyhelminthes, rotiferans, some of the anarians and cephalochordatans. And now coming on to the next one, antennal gland is present mostly in the arthropodon crustaceans present in the water like prawns. So where the excretion is done through the glands which are present in the antennal, and they look mostly in the green in color, so they are called as antennal green glands. And the next one is the Malpighian tubules. In most of the cockroaches and most of the insects, this type of excretory organs are present, which was discovered by Malpighi So they were named after him as a Malpighian corpuscles tubules. Now coming on to the next one, nephridia. Nephridia is present in most of the earthworm, and you can see that two types of nephridia are present. One is called as a pharyngeal, which is present in the pharyngeal portion, and integumentary. The human kidney nephron is a higher version, that is the updated version of this protonephridia. And now coming on to the kidney. Kidney is a functional and structural unit of elimination in the body from the by filtration of blood from the body. Generally, when you see the kidney dimensions, 10 into 5 into 4 centimeters is the dimension, that is length into breadth into thickness. It ranges from the 10 to 12, 5 to 6 and 2 to 4 centimeters in L into B into T. And when you weigh the human kidney, we are learning about the human kidney. So human kidney weighs approximately between the weight of 120 grams to 170 grams, and it is present in the last thoracic, 12th thoracic cavity to the 3rd lumbar cavity in the dorsal side. And you can see that when you see onto the kidney is bean shaped or bean shaped. Kidney is having a shape called bean like structure, and you can see that the anterior portion of the kidney having a notch called as hilum. Hilum consists of a notch, ureter, nerves and blood vessels enter into the kidney through this hilum. And the next portion to that is called as renal pelvis, which is a broad funnel shaped structure just below the hilum, which is coming out of the kidney, which collects the urine from the whatever the filtrate from the kidney. And you can see that nephrons are present. Approximately 1.2 million number of nephrons are present in each kidney, and they are of having two parts. One is the upper part, cup-shaped part called as the glomerulus, and the lower part, tubular part called as the renal tubule or nephron tubule. The glomerulus will be made up of 3 layers. So you can see that when you come onto the kidney is having a C-shaped or curve shaped on front side where the notch is present, that is called hilum, and the outer part where the kidney is present, outer part which is the portion called as a dark colored portion called as a cortex region, and the inner part called as medulla region. Pyramid is present into the medulla region, part of the medulla, where all the collecting ducts unite and form the renal pyramids, where renal pyramids pour their secretions or pour their filtrate into the renal pelvis. And you can see that in the case of between the two pyramids of the renal pyramids, which are generally 9 to 12, you can see a columns called as columns of Bettini, which are the cortical extensions between the medullary pyramids, and you can see that these columns of Bettini will be supported with certain other globular network, and you can see that each nephron consists of 2 parts. Already we have discussed that the renal, the cup portion and the tube portion. So the glomerulus, which is originating from the renal artery and formed as a afferent arteriole, will be moving into the glomerulus, and each glomerulus will be in connecting that such a way that you can see that the part of the after the Bowman's capsule, the first one is called as a proximal convoluted tubule or first convoluted tubule, connected to the next part called as a loop of Renal, where it is coming down from the proximal convoluted tubule, it is called as descending loop of Renal, and when it is going up forming a hairpin like structure called as a ascending loop of Renal, and which is connected to the distal convoluted tubule or the proximal convoluted tubule, and which is the proximal convoluted tubule is connected to the collecting duct. And generally we can see that when you see the shape of a Henel's loop, it is hairpin shaped, and the Henel's loop is meant for the concentration of the urine in the kidneys. And you can also see that there are two types of nephrons. One is based on the presence of their location and their functionality. You can see that which are morely restricted to the cortical region and which are 20% and they will be having small Henle loop or no Henle loop. Those are called as cortical nephrons, and others are called as juxtamedullary nephrons or medullary nephrons. So this will be 80%. And around these nephrons, you can see that a counter current or opposite running blood vessel which is formed from the efferent arteriole coming out of the glomerulus will again collect the blood in the reverse direction to the movement of nephron filtrate, called as the movement is called as counter current movement. And the shape of the vasa recta is U-shaped and the shape of the Helen's loop is almost V-shaped. And you can see that other capillary network of peritubular network is also present, and generally in the human whatever the amount of blood pumped by the heart, approximately 1/5 amount of the blood is filtered by the kidneys only. So per minute, that is, you can see that 1/5 into 5000 ml approximately per minute, it pumps 5040 ml of blood. So in that approximately between 1000 to 1100 ml of blood is filtered by the kidneys. And there are three important processes taking place in the kidneys. First one is the filtration, followed by the reabsorption, followed by the secretion. Filtration, the blood is filtrated, and as the blood is filtering to the glomerulus, and all the glomerulus, it is also called as the rate at the filtration takes place in the kidneys. In each of the glomerulus is called as GFR. GFR of the human kidney is 125 ml per minute per time. That is, two kidneys will filter that much amount of glomerulus, so it is also called as GFR. And coming to the reabsorption, so 99% of the filtrate what is done will be reabsorbed, and only 1% is converted. That is approximately in a day 180 liters of urine, 180 liters of blood is filtered, and only 1.5 ml of urine is passed per the day from the human body. Excess to that is more amount of urine, rather 6 to 8 liters to 15 liters is called as glycosuria, is also called as diabetes insipidus. That is because of the failure of vasopressin or ADH. And reabsorption is of two types. One is called as active reabsorption, one is called passive. Active means with the presence of energy, passive means without the need of energy. In the active, you can see that you can remember NAG, okay, which is the glucose, amino acids and sodium ions are passed out of the kidney through the active transport, and passive transport, NM, that is van, water and the nitrogenous waste will be absorbed by this process without the need of energy. And coming to the secretions, so during this filtration and reabsorption, some amount of will be pushed out by secreting certain substances like creatinine, H plus ions, K plus ions, NH3, so which is called as a CH, chicken. Secretion is chicken. And you can also see that when you talk about the glomerulus, it is having three layers. One is called as the endothelium of the blood vessels, epithelium of the blood capillaries, and also basement membrane present between these two forms the glomerulus. So these are the three layers of the glomerulus. And you can see that filtration is a non-selective process generally performed by glomerulus by a pressure called as glomerular capillaries blood pressure, and approximately 1200 ml of blood per minute is filtered by all the glomerulus, that is 125 ml per minute in blood capillaries in the blood Bowman's capsule per minute. So your glomerular filtration rate, already we know that it is 125 ml per minute. that is 180 liters per day, and generally 99% of the blood is reabsorbed. And you can also see that only proximal convoluted tubule, there is a brush border cells, which is very important point, mostly asked many of the times. And the concentration of the urine takes place in the cortex and medulla of the Helen's loop. Generally, from here to here, the osmolarity differs, and in the cortex, it is approximately 3000 ml osmoliters per liter. But here, when it so as the pressure increases, also the reabsorption as well as the concentration increases. And generally, from here to here, 4 times the concentration of the urine increases. And when you come on to the collecting duct, here large amount of, that is whatever, here also selective reabsorption takes place, and large amount of water is reabsorbed in the collecting duct. And you can see that in the case of the vasa recta, it is a selective secretion, and which is a counter current mechanism. And you can see that concentration, already we have discussed that the Helen's loop and the vasa recta are responsible for the concentration of the urine, and generally acidification of the urine in the cortex is done by the phosphoric acid. And when there is a blood pressure increase, heart releases vasodilator, atrial natriuretic factor, which is responsible for the controlling, that is vasodilation or vasoconstriction, is under the control of renin, aldose, angiotensin, aldosterone and mechanism. So here microtusion, that is the feeling of passing out of urine from the body because of the pain is called as microtusion. This signal is generated by the brain itself, and this is in control with the stretch receptors present on the bladder, responsible, which is taken the signals from the central nervous system. And you can see that excretion is done by the kidney. Apart from the kidney, which is the major functionality, excretion is also done by the kidney, liver and skin. And the kidneys, already you know, liver is meant for digesting one form to the other form, like the digestion of the blood into other simpler components. And excessive presence of urea in the urine is called, in the body is called as uremia condition. And you can also see that there is sometimes the disorder, coming to the disorders of the kidneys, you can see that kidney stones are also present. This is called as renal calculi condition. And the inflammation of glomerulus because of the bacterial, viral or other infections is called as glomerulonephritis. In the case of a patient suffering from the more of the, having more because of kidney failure, they can to lead a successful life. So to elongate a life, a machine called as dialysis, which was discovered by William J. Kolff, which is called as the artificial kidney or dialysis machine, and the permanent solution for this kidney failure is kidney transplantation, which was performed by Charles Affengal in the Washington in the year 1954. Thank you.